Assalamualaikum and hello Dr. Izina. I am Aisha from Group 6. So today, me and my friends would like to present about the experiment determination of carbohydrate and lipid digestions. So firstly, I would like to introduce my group members, which are Tunku Farid bin Tunku Fazli, me, Siti Hajar Aisyah binti Khairul Anwar, Khairiyah binti Jasmi, and also Nur Shahira binti Shamsuddin.
The objective of this experiment is to examine the function of common enzyme in the digestion of starch and protein. Uh, and the second one is to determine the factors that affecting enzyme activity. First experiment is the digestion of starch. The methodology is first we need to prepare 5 pair of test tube. First pair contained 4 ml glucose only and was labeled as A1 and A2. The second pair is contained 4 ml of starch only and was labeled as B1 and B2. The third one is 4 ml of untreated amylase and was labeled as C1 as C2. The fourth one contained 2 ml of starch and 2 ml untreated amylase. And the last one is 2 ml of starch and 2 ml of acidified amylase and was labeled as E1 and E2. After that, all test tube was well shaken and was left for 10 minutes. Uh, after 10 minutes, uh, 0.5 ml of iodine solution was added into test tube A1 until E1. It was shaked well and observation was recorded. And at the same time, 0.5 ml of Benedict reagent was added into test tube A2 until E2 and was shaked well and incubated in boiling water bath for 100 degrees Celsius for 5 minutes, and after that observation was recorded, and the observation was recorded in the table of the result.
Hi, it's me Aisha again. So, I would like to present about the result for this experiment. Firstly, from the pair of test tube A, which contained only 4 ml glucose, it shows a clear brown or no change in color for iodine test. Meanwhile, it shows a brick red or dark red color in Benedict test. So, the color change in Benedict test shows that glucose is present. From the pair of test tube B, which contained only 4 ml of starch, the test tube shows a dark blue color when it is tested on iodine test, and it shows a no change in color when it is tested in Benedict test. This shows that starch is present. For the pair of test tube C, which contained only 4 ml of untreated amylase, shows a clear brown color or no change when it is tested in iodine test, and it shows a light orange color when it is tested in Benedict test. These changes in color conclude that glucose is present. From the pair of test tube D, which contain 2 ml of starch and 2 ml of untreated amylase, the result shows a dark blue color when it is tested in iodine test, and it shows orange color when it is tested in Benedict test. These changes in color shows that starch is present and so is glucose. From the pair of test tube A, which contain 2 ml of starch and 2 ml of acidified amylase, the result shows a dark blue color when it is tested with iodine, and it shows a light orange color when it is tested to Benedict solution. These changes in color also show that starch is present and so is glucose. So let me present about the discussion part of this experiment. Each of the pairs of test tubes are tested with iodine and Benedict solution for each one of the pairs. Iodine is used for testing the presence of starch, which turn into a blue-black pigment when starch is present. Benedict solution is used for testing the presence of reducing sugar, which turns a blue Benedict solution color into a reddish pigment. Later, the test tube that contains Benedict solution will be immersed in a water bath of 100 Celsius for 10 minutes, and then the results are then recorded.
Right? So these are the examples of when there's a presence of reducing sugar in the Benedict's test, right? So this is a high concentration or high presence of reducing sugars, so that's why it turns into a really red color. While if there is no presence of any reducing sugar, it remains into this blue, light blue color. Right? For the iodine test, it turns in if there's a high presence of starch, it turns into a really dark blue-black pigment. While if there is a low presence of starch, it turns into a lighter color than. So for pair A, 4 milliliters of glucose only is tested with the test, the iodine and Benedict test. Where the iodine test, it remains clear brown from its initial and final stage, while in the Benedict's test, it turns into a brick red pigment. So from here, we can infer that glucose is present. As to the Benedict's test, it turns from the light blue color it is to a brick red pigment. So glucose is present while starch is absent, as the color of iodine, when tested with the iodine test, it remains the same. There's no change. For pair B, 4 milliliters of starch only is tested, right? So in the iodine test, it turns into a blue-black pigment, while for the Benedict's test, there is no change. It remains light blue, right? So from here, we can infer that glucose is absent, as there's no change in the Benedict's test, while starch is present, as it turns the iodine into a blue-black pigment. Right? So in pair C, 4 milliliters of untreated amylase is used. Right? For the iodine test, it turns into a clear brown, and the Benedict's test, it turns into orange. Glucose is absent and starch is absent. There is a bit of a wrong result that we got in a group, as there's some contamination with some other substances that may have caused this positive reading of reducing sugars in the amylase. Right? So for pair D, 2 milliliters of untreated amylase and 2 milliliters of starch is used. So in the iodine test, it turns into a blue-black pigment, and in the Benedict's test, it turns orange, right? So from here, you can infer that glucose is present and starch is present, right? So amylase breaks down starch, a complex sugar, into a more simple sugar, glucose. That's why it turns this into orange. While there's still some starch left, as the digestion hasn't completely taken place. That is why even after the water bath, when tested, when iodine is present, it still turns into a blue-black color. Right. For pair E, 2 milliliters of acidified amylase and 2 milliliters of starch is tested. So the iodine test, it turns into a blue-black pigment, while for the Benedict's test, it turns into orange. So glucose is present and starch is present as well. So it's the same as pair D. Amylase breaks the starch down from a complex sugar into a more simple sugar, glucose. That's why it turns into this orange color when tested with the Benedict solution. Well, it still when tested with the iodine solution, it still remains. It changes into a blue-black pigment.
Next, we will move to the experiment 4.3, which is digestion of lipid. The enzyme used in this experiment is lipase. So lipase will break down lipid into glycerols and fatty acids. So in this experiment, we use phenolphthalein to indicate the presence of fatty acids. So, okay, next for methodology, we have 4 test tubes. So the first one, we put 2 ml of lipase and 4 ml of bile. So what is the function of bile? Bile is to break down fat into smaller molecules, so it increases the surface area, and so lipase will work on it quickly. And then we put 3 drops of phenolphthalein and sodium hydroxide. So why we need to put sodium hydroxide? Because we want to make the initial solution in all test tube A until D in alkaline state. So then we put 3 drop of vegetable oil, and we we put the test tube in incubator, and the temperature for incubator is 37 degree. Okay, for test tube B, we it is same as test tube A, but we don't put bulb. Okay, for test tube C, we want construct the role of lipase, so we don't put the lipase in test tube C, and but the procedure is still same. For the test tube D, we want to know the effect of temperature, so we put the test tube D with similar procedure as the test tube A, but we put the test tube D in room temperature. Okay, for the result, there are some errors in this result, but it will be discussed by Tengku in the discussion later. So initial color of test tube A is is pink to red, and the final is green. The color turned to green because of bile, so we consider the color is colorless. So that means that the fatty acid are present. Okay, next, for test tube B, we don't put bile in this test tube. So the initial color is pink, and final remains same. Means that the fatty acid are absent for this test tube. Next, test tube C, we don't put lipase in this test tube. So the initial color is red to pink, and the final is dark pink. Means that the fatty acids are absent. Okay, and for the last test tube, we put lipase in bulb, but we put test tube D in room temperature. So uh, the initial color is red to pink color, and the final is brown. So means that the fatty acids are absent. Okay, that's all. That's all from me. Thank you.
Right, I'll be presenting the discussion part for the digestion of lipid or experiment 4.3. So the lipid here used is vegetable oil, where 3 or so droplets are used for each test tube. Bile is used for the emulsion of lipid. This basically means it breaks it down into smaller droplets, therefore increasing its surface area, which is important as it increases the rate of reaction between the lipid and its enzyme, right? So sodium hydroxide is used to control the alkalinity of the mixture as it's an alkaline solution, right? Phenolphthalein is used as an indicator of that, right? Phenolphthalein changes color from a colorless solution into a bright pink pigment in a high alkaline environment. Okay. So lipase is the enzyme used for the digestion of lipids in this experiment. So from then we can know that the enzyme is at its highest rate of reaction in a high alkaline or high pH environment, right? As it can be found mainly in the duodenum, a very high pH environment in our digestive tract. Right, for test tube A, 2 milliliters of lipase and 4 milliliters of bile is the solution used. It is left in a 37 degrees Celsius water bath for an hour, and the results are that the lipase is able to digest the lipid. Right, so the final result is colorless, or this bile color that can be ignored. So basically, it is colorless. So we can conclude that fatty acids are present in test tube A. Right. So for test tube B, 2 milliliters of lipase is mixed with 4 milliliters of distilled water. And the solution is left at 37 degrees Celsius water bath for 1 hour as well. So over here, we can see that lipase is not able to digest the lipid, right? And the final result is pink. Fatty acids are absent. For test tube C, 2 milliliters of distilled water is mixed with 4 milliliters of bile along with the droplets of oil. at 37 degrees Celsius water bath for 1 hour. Right. So the results are the lipase is not able to digest the lipid. The final result is still pink, and the fatty acids are absent. Okay. So for test tube D, 2 milliliters of lipase is mixed with 4 milliliters of bile and left at room temperature for 1 hour. So the results that we got from this test tube is that lipase is not able to digest the lipid, and the final result is pink. Right? The fatty acids are absent. Right. So there are a few things that we can improve that can be improved in the experiment that we have conducted. So the solution in the test tube should have a really pink pigment, which we can't really see as to example 1.0 here. The solution should be a brighter pink compared to this pigment here, which is too dark. So therefore, it's in a lower or less alkaline environment or solution where it decreases the rate of reaction of the enzyme lipase here used as it thrives in a higher pH value, right? So the reason being the enzyme used is at the highest rate of reaction at a high pH, right? Lipase is found in the duodenum where it lives in a really, or it stays in a really high pH environment. So it should be kept there. So it should be a brighter pink than this. Phenolphthalein turns pink in alkaline environments. So therefore, this should be really pink. And this can be achieved by adding more sodium hydroxide to the solution. So adding more sodium hydroxide increases its alkalinity or pH value, therefore giving the enzyme a more high alkaline environment.